Hello everyone and welcome to today's video. In today's video, we're going to be discussing the Oxford AstraZeneca vaccine technology, answering a few questions, and in general, just understanding how this vaccine really works. So first of all, we have to understand that there are two major components for this vaccine. Number one is the vector that is holding the DNA and the DNA itself that is going to be coding for the protein that is going to be essentially giving our immune system a heads up regarding the SARS-CoV-2 spike proteins. We will get to that at the end. However, let's go back for our vector. Our vector is, in this case, the chimpanzee adenovirus that is going to be harboring our S protein coding DNA, and they are going to be present in the vials that we're going to be injecting into our arms. Now, since this adenovirus is going to be harboring this DNA, it is going to be entering through our cell membrane into our cytoplasm and then injecting our DNA, in this case, the region or the DNA gene that is going to be coding for the spike protein. It's going to be injecting it into the nucleus. Now, it's very important to actually mention that there is going to be no incorporation of this gene, this DNA fragment, into our genome. It's simply going to be using specific enzymes that are present within the nucleus in order to get converted from DNA to mRNA. And after that, it simply follows the exact same route that the Pfizer, BioNTech, and Moderna vaccines take. I'll try to make it as short as possible. So this mRNA now is going to be transported to the cytoplasm, where it's going to be converted into proteins via translation. Then these proteins are going to be expressed on the surface of the MHC class 2, where a chain of reactions are going to be taking place, where the T helper cells are going to be recognizing these proteins, then releasing cytokines, triggering the release of antibodies by plasma cells that are going to be playing major roles in the neutralization of SARS-CoV-2 upon entry. Also, we're going to be having the presentation of the proteins that we mentioned here onto MHC class 1 proteins or receptors that are present on the surface of the cell membrane of the host. This is going to be triggering the cytotoxic T cells or the CD8 positive cells to release destructive enzymes that are going to be cleaving and destroying cells that are infected with SARS-CoV-2. So this concludes this video. Thank you for listening. This has been Ali from B Biology.